Wild-type bees that are more sensitive to sugar were found to demonstrate a pollen foraging bias, an early onset of foraging. They collected lower quality nectar and would even collect water. This is a phenotypic architecture in wild-type bees that are more sensitive to sugar that is equal to the high-strain bees. High-strain bees are more sensitive to sugar. So we're building, this is an architecture of behavior that is associated with high and low strain bees, and also associated with bees on the basis of their sensitivity to sugar. So we're showing an architecture. I had an undergraduate student working in my lab, and she was doing some dissections of bees. And she came in one day and she said, you know what? The high-strain bees have bigger ovaries than the low-strain bees. And so this was like remarkable. We tested it, and we found out that in fact they did have more ovarials per ovary. So these are the ovaries, and the ovaries consist of filaments called ovarials. And each one of those ovarials is an assembly line for making eggs. And so the ovaries, they get activated and they swell, and then they start absorbing vitellogenin from the blood. And the vitellogenin is an egg yolk protein that they then make into eggs. And so honeybees, workers, can make eggs. So what she found was that the ovaries of the high-strain bees were activated. They were what we call vitellogenic. They're absorbing vitellogenin titers for making eggs. And so we tested them in different ways. We also found that they were, if you remove the queen from a colony, the workers will develop their ovaries, they'll make eggs, and they'll lay eggs. They become laying workers. They develop faster into laying workers than did bees from the low strain. So if you tested them together and put them in a colony, removed a queen, and they're for the same age individuals, the high strain workers would more rapidly develop into egg layers. So they're overall more reproductively activated when they're very young than are the low strain bees. So then we tested that with wild type bees to see, okay, if you look at wild type bees and we look at their ovaries, what we find is that bees that have more ovarials, in other words, you test them and you dissect them, if they have more ovarials, they're more sensitive to sugar. They forage earlier in life. They show a pollen foraging bias. They collect nectar with less sugar. They'll accept the lower quality sugar. That's associated with being more sensitive to sucrose. They're more likely to forage successfully. And they have higher vitellogenin titers floating in their blood. So they're more activated reproductively than are, if they have more ovarials, than are those wild-type bees in the same colony that have less activated ovaries. So we've got this connection between ovaries and behavior. So collectively, we looked at a lot of different things. We looked at gene expression for specific genes that we suspected might be involved in vitellogenesis and reproductive signaling. And we looked at many different kinds of foraging behavior and hormone levels. And what we found was again a phenotypic architecture. It's like that dog balloon that we had. The things are related to each other. The things that stood out was the response to sugar was a very linked to many, many things. And that's probably because of the tuning of the sensory response system that's going on when individuals are reproductively activated. And those with more ovarials, they link to all these more of these traits as well. So these are kind of key things. The ovarials, we thought the ovaries are somehow involved in signaling a lot of things that result in tuning the sensory system and affecting behavior. There's also a genetic architecture. Genes don't work in isolation. Just like phenotypes aren't isolated traits, they're linked together in an architecture. The genetics are too. When you look at genes, take a complement of genes, you'll find gene networks are out there and working. So you have a collection of genes here that are working together and they're affecting each other, and you have another collection over here, and these may be somehow related to different kinds of traits. The things that they're regulating or building at any given time might involve these different networks of genes. They don't work in isolation, they're working together. We also found that there's a genetic architecture associated with explaining the differences between our pollen hoarding, our high pollen hoarding lines and our low pollen hoarding lines. We were able to map through the process of QTL mapping. We were able to map regions of the honeybee genome, reflecting differences between our high strain and our low strain. We were able to identify regions that contain genes that were affecting the phenotypes that we were measuring that were varying between the high and low strain. So we did a QTL mapping project. What we found when we did the QTL mapping project is this architecture where we identified 4 regions of the genome that explained a lot of the behavior that we were looking at: pollen and nectar load, nectar concentration, sucrose response, and foraging age. So this was part of our phenotypic architecture. So we have a genetic architecture here that is intimately linked and networked with our phenotypic architecture. And the genes are interacting with each other in a highly interactive way, and they're affecting multiple traits. So these 2 architectures are working together.